Okay, so hello, fellow brain cells, and today's episode of This Week in Neuroscience, we're going to talk about this new paper uh, about AgRP neuron hyperactivity, how it drives hyperglycemia in a mouse model of type 2 diabetes. Okay, so um, as it states, work in these recent decades implicates the brain both in pathogenesis, I'm not sure how it's pronounced, of type 2 diabetes and as a target for its treatment. And specific neuronal subsets responsible for effects of for these kinds of effects are unknown, but mm, this current work investigates the role played by a specific subset of neurons, who are AgRP neurons, who are located in the hypothalamic arcuate nucleus, which, as I have outlined here in the notes, it acts as a key control center for appetite and energy balance, hormone regulation, reproduction and growth, and metabolic processes, and it is located at the base of the hypothalamus near the third ventricle. Okay, so about this leptin deficient LEP-OB mice. Now, what the hell does this mean? So this is called a mouse model. Uh, these mice are genetically modified to lack leptin, which is a hormone made by fat cells. It signals to the brain that the body has enough energy stored, like for body fat, and without leptin, these mice overeat, which is called hyperphagia. They become obese and they develop insulin resistance and high blood glucose, which is basically type 2 diabetes. Um, so, uh, the same authors found that in 2016 that a single intracerebroventricular, uh, which is which means placing the injection into the fluid-filled brain ventricles. This injection of fibroblast growth factor 1, which is a protein involved in cell growth and metabolism, um, a single injection of that can induce diabetes remission lasting months. In a series of subsequent studies, identified the AgRP neuron inhibition as a potential mediator of this anti-diabetic effect, and the effect of this fibroblast growth factor 1 was localized to the um, arcuate nucleus. Yes, arcuate nucleus. I can't seem to remember that. And shown to require intact melanocortin 4 receptor signaling, which is, I have it here. This is a receptor that reduces appetite and increases energy use, and it's important for controlling body weight and glucose. So this receptor is blocked by AgRP neuron activation, and since AgRP neurons are both hyperactive and inhibited in a prolonged manner following this injection, the authors hypothesized the key role for durable inhibition of these neurons in the sustained anti-diabetic effect elicited by this intracerebroventricular, hope I'm saying that right, FGF1 injection. And in support of this hypothesis, they report that permanent AGRP neuron inactivation recapitulates the sustained diabetes remission induced by this injection in these mice, genetically modified mice. And this effect has occurred despite having no detectable impact on food intake, energy expenditure, body weight, or body fat mass. Their findings identify AGRP neurons as key drivers of hyperglycemia, but not obesity, at least in that mouse model, that type 2 diabetes model. Uh, So, sorry. So they immediately jump to the results, and I want to emphasize on this particular really difficult sentence that took me a bit. So to permanently inactivate these particular neurons, AgRP neurons, a Cre-inducible tetanus toxin GFP AAV vector was microinjected into the ARC of lab OBOB AgRP CRE mice. Now, that's a mouthful. That's quite a mouthful. Now, what the hell does this mean? Wow. Cre-inducible basically means that this kind of DNA only turns on if the cell has a special enzyme called CRE, CRE. And tetanus, we know about tetanus toxin GFP. Now the GFP is green fluorescent protein, and which basically makes the cell glow green under a microscope so that scientists can see it. The scientists can see it. AAV vector. AAV is basically an adeno-associated virus used to deliver genes into the brain cells. So this is the virus that transports the DNA, which only turns on if the cell has a special enzyme called CRE. And actually, what is tetanus toxin, TTX, tetanus toxic light chain, which is a modified form of the tetanus toxin, but it doesn't cause disease. It only blocks the neuron from sending signals by stopping neurotransmitter release. So basically, this is the inhibition. This is how they achieved. Now we already talked about the ARC. Okay, I mentioned all of that. So that sentence is finally at last complete. So there was also a control group, of course, and control mice received an injection of this adeno-associated virus, which contained only Cre-inducible GFP reporter. So only this green fluorescent protein that's activated only when there's a special enzyme called CRE. So 3 mice were excluded on the basis of inaccurate bilateral targeting. Let me just... bilateral targeting. Let me just check. What does this mean? Must have forgot. Bilateral versus unilateral target. Mode destructor. Okay, bilateral target. Under bilateral target presentation, larger remote distractor effect. This, yes, I guessed it correctly. Remote distractor. Anyways, major curve for central distractors, whereas under unilateral presentation, the pattern reversed. Okay, this just uses the word, it does not explain it. Bilateral. Wait, what is... Okay, maybe if I just say the word bilateral. Am I a dumbass, or am I not the only one that doesn't get this? Bilateral version. Okay, we read that. Bilateral deep brain stimulation of the subthalamic nucleus. Bilateral targeting. What is bilateral targeting? Let's see the AI response. I don't know anymore. Whoops. What did I click on? So bilateral targeting in neuroscience related fields refers to simultaneously targeting or stimulating specific brain regions on both sides of the brain. Oh, so I guessed it in my head, right? I should trust myself more. Maybe. Okay. They did that inaccurately, which of course fine to they better be excluded, of course. So GFP controls, we already covered that. Okay. So this is just the mean. So the mean blood glucose level fell from 230 milligrams per deciliter to 140 milligrams per deciliter, and the main daily blood glucose level diabetic controls remained well above 200 milligrams per deciliter for the entire 10-week period of the observation. So this is a 10-week period of observation, and their results lasted 10 weeks, which is like gosh, almost 4 months, like 3 months and a half. Oh. So there in the requirements they state that a remission, a diabetic remission, is considered maintained blood glucose level under 200 milligrams per deciliter. So, of course, that's good. And as we point out, this outcome cannot be explained by reductions of either food intake or body weight, as neither parameter differs significantly between groups over the 10-week course of the study. And also, AgRP neuron inactivation had absolutely no effect on body composition, energy expenditure, respiratory quotient, or ambulatory activity. Actually, what is this respiratory quotient? I got curious. Didn't look it up. Respiratory quotient. Okay, this should be easier to find. I feel like. Okay, the respiratory quotient, or the respiratory ratio, is the volume of carbon dioxide released over the volume of oxygen absorbed during respiration. Oh, alright, okay, good. Ambulatory activity. I also am not sure what this is, so let's look it up. This is APA. Ambulatory activity monitoring defines the measurement strategy to assess physical activity, posture, and movement patterns. So, general physical activity is what I'm getting. Defined as direct measurements of the amount of walking and or steps taken over time. Yeah, maybe it has different operational definitions for the different studies, but in general it's activity that's relevant to the study. So what I'm guessing here, this means, okay, we covered that. They repeat themselves a bit in some sections. Okay, so potential mechanisms mediating the effect of AgRP neuron inactivation to normalize diabetic hyperglycemia, which is high level of blood sugar. Okay, so they examined liver fat and glycogen content as well as hepatic glucoregulatory gene expression. What is that mouthful? Wait, does this have sources? Journal, Arizona Cancer Center, NIH. Okay, then this should okay, so these are NIH is good, Journal of Endocrinology is also good. Okay, so we can trust this AI right now. Hepatic glucoregulatory gene expression, referring to the genes involved in regulating glucose metabolism in the liver, is tightly regulated by various factors, including hormones, nutrients, and circadian rhythms. Key genes include progenic enzymes. These genes are influenced by factors like glucagon, insulin, and fatty acids, as well as the circadian clock. Circadian clock. Almost read. So, while reduced liver fat content is often associated with improvements in insulin sensitivity and glycemic control, no significant differences were observed between the TTX, tetanus toxic light chain, GFP treated mice, and the controls. So, similarly, neither hepatic expression of the key glucoregulatory enzyme glucokinase or the gluconeogenic genes. I swear I read this, but I read this article, but what is this word? Phosphoenolpyruvate carboxykinase and glucose 6 phosphatase differed between groups. In contrast, liver glycogen content was significantly increased, as has been observed following this injection in the genetically modified mice. So, let's see. Okay. Here, I believe we can see blood glucose. We can see the more meaningful change in the TTX condition. Body weight. It's basically no difference, as previously stated. Food intake, nearly no difference. Even in some cases, the TTX is higher, which is interesting. But again, they said that it was not statistically significant, so yeah. Okay, there was also some here. Heat production, also no significant difference. Also, we can see it as body weight. What is RQ? Oh, the respiratory quotient. Yeah. Oh. Okay, heat production activity. Okay, yeah, the controls had even slightly higher here and here, but again, only 2 cases. Let's see the others. Blood glucose, we can see this absolute beauty of a result. Absolute beauty. Plasma insulin as well, which is interesting. What is plasma insulin? What is the difference between this and other insulin? What are the sources? Whoops. Medscape reference, sciencedirect.com, NIH. Okay, these two are good. But this, what is this? Medscape. What? This site is intended for healthcare professionals. Huh. Okay, so this is news about articles, I'm guessing. Okay. So, again, Plasma, we trust the AI since it has credible sources. Plasma insulin, also known as serum insulin, is a hormone produced by the pancreas that plays a crucial role in regulating blood sugar levels by facilitating glucose uptake into cells. And it's measured in the blood to assess insulin sensitivity, resistance. It can be used to identify causes of hypoglycemia, not hyper, but hypoglycemia. Okay. But this is what it's all about. This is what it's all about in a single graph. Let's see, liver glycogen, liver fat, liver mRNA, fold change. Oh, these were the enzymes. Okay, so I skipped. By the way, I highlighted. I had highlighted things throughout the entire article, and then and it was saving in the cloud or whatever. Apparently it was saving on like every highlight. It was saving, and then I go back in Mendeley and everything is wiped clean. So that's why I am doing it this way today. Decided to try this out, see how it goes. Okay, so let's see. So during fasting, HPA axis activation is causally linked to AgRP neuron activation. Just a second here. Discussion. Okay, I think because the discussion is like the biggest part of this paper. Yes, it is the absolute biggest part. As you can see, this is all of this is is a discussion. Yep, from here. Okay, so let's summarize. So AgRP neuron hyperactivity alone is enough to maintain high blood sugar in this, at least, type 2 diabetes model, and silencing them or inhibiting them brings blood sugar down, even without low weight loss or lower food intake. This mirrors the effect of the fibroblast growth factor 1, but it may have unique advantages like reducing stress hormones, and suggests a brain-specific target for diabetes therapy that spares appetite and energy balance. But it does raise big questions like, why does this work for glucose but not for obesity? There's obviously some other underlying mechanism for other symptoms and mechanisms involved in type 2 diabetes. And also, could current drugs like GLP-1 or GLP agonists work partly by targeting AGRP neurons, which is, I think, in itself grounds for future research. And other subsets of AGRP neurons may be responsible for different metabolic effects. Also, a very, very interesting research prospect, I think. Okay, and what I'm interested actually, would you take a brain targeted treatment if it meant normal blood sugar but no weight loss? And do you think what do you think how much potential do you think neuron targeting has in treating diabetes? Or and is it possible we're actually underestimating the role of the brain in such seemingly purely physical diseases? So I'd love to know what you think. Today's episode was a bit short but sweet, and but it is the article itself is not that long. Like, you may think it's 10 page. It is 10 pages, but there's also so many graphs in here, so it's not really that long, but still it is quite important. And so that was AgRP neuron hyperactivity drives hyperglycemia in a mouse model of type 2 diabetes. This week in Neuroscience, out.